All right, today we're going to break down how two very different COVID-19 vaccine technologies were engineered to work with our immune system. We're going to explore their unique strategies and, well, some pretty surprising and unintended consequences. So, let's just dive right in. So, here's the core idea of this whole analysis, laid out right here. You've got the Western mRNA platforms, Pfizer and Moderna on one side, and on the other, this PLA/AMMS research platform. Now, even though their designs are completely different, this analysis suggests they might actually end up in the same dangerous place with two shared outcomes. First, suppressing our body's crucial antiviral interferon response, and second, accidentally triggering autoimmunity through something called molecular mimicry.
Okay, so let's kick things off with part one, which is all about that Western mRNA platform. The key concept here, as the source calls it, is engineering chemical evasion. So the big challenge for this tech was a huge one. How do you get this synthetic messenger RNA into our cells so they can make the spike protein without our immune system immediately freaking out, seeing it as an invader and just destroying it? Well, the solution was this three-part strategy: a lipid nanoparticle to carry the message, a special stabilized design for the spike protein, and a really clever chemical trick to sneak past our body's immune sensors.
And that chemical trick? It all comes down to this one molecule, N1-methyl pseudouridine. Basically, it's a synthetic modified version of a natural building block in our RNA. Its all-purpose in this vaccine design is to act as a disguise, like an invisibility cloak.
So, how does this chemical cloak actually work? You see, normally our immune sensors would spot foreign RNA and sound a massive alarm. That's the Type I Interferon signal, to get rid of it. But by swapping in N1-methyl pseudouridine, the vaccine's RNA basically puts on this cloak. It slips right past those sensors, which means that critical antiviral alarm gets suppressed. This allows our cells to produce a lot of the spike protein for a longer period of time.
Okay, so the package is cloaked. It gets past security. That makes sense. But that leads to the next huge question. What about the payload itself? What about the full-length spike protein that our cells are now being told to churn out?
And that brings us to a really tricky problem called molecular mimicry. It turns out the spike protein our cells are producing contains this tiny little sequence of just 5 amino acids, RLFRK. Now the issue, as this analysis points out, is that this little viral sequence looks alarmingly similar to some of our own critical human proteins, including one that's key to our interferon response and another that's vital for our nervous system.
So, what happens when the immune system gets fooled by this lookalike? Well, the first potential risk identified here is a big one. The immune system might start attacking its own antiviral defenses.
Okay, check out this potential vicious cycle they describe. A T cell gets trained to hunt down that viral RLFRK sequence, but because of this molecular mimicry, it might get confused and attack our own PPI-P5K2 protein by mistake. This messes up the entire machinery we need to produce that Type I Interferon, our main defense against viruses. The end result could be a kind of autoimmune acquired immunodeficiency, where the very response to the vaccine could, paradoxically, weaken the system we need to fight off future viruses.
But the risk doesn't stop with our antiviral defenses. The analysis points to a second potential target for this case of mistaken identity, our own central nervous system. That same viral RLFRK sequence also looks like a protein called Tenurin-4, which is absolutely essential for myelin. That's the protective insulation around our nerve cells. An autoimmune attack on TENM4 is linked to demyelinating disorders. So the source suggests this molecular mimicry could be a possible mechanical explanation for some of the clinical reports of conditions like transverse myelitis or multiple sclerosis that have been observed after vaccination.
All right, let's switch gears to part 2 and see where this is all going. We're now looking at the PLA/AMMS platform, which took a totally different route. Not chemical evasion, but engineering functional suppression.
So instead of using the whole spike protein in a chemical cloak, this research focused on just a small piece of the virus, the receptor binding domain, or RBD. And rather than hiding from the immune system, this strategy actually used a built-in feature of the spike protein itself to actively shut down the immune response.
So here's the play-by-play on how that works. The spike RBD has this natural sequence called the RGD motif. This little sequence can actually bind to our own cells, hijacking them and forcing them to release a powerful immunosuppressive molecule called TGF-beta. This TGF-beta then goes and directly shuts down the production of Type I interferons, getting to the exact same result as the mRNA platform, but by using a biological pathway instead of a chemical trick.
And this leads to a really strange twist in the story. As the source puts it, a strategy intended to enhance vaccine efficacy created a concentrated autoimmune risk. See, this platform used a strategy they call immunofocusing. By using only the most important part of the virus, the RBD, they could focus the entire immune attack right on that one target for maximum power. But here's the incredible paradox. The RBD is also exactly where that high-risk RLFRK sequence is located. So the unintended consequence was that the vaccine was basically optimized to create a super powerful, super specific immune response against the very sequence that mimics our own human proteins. And this is where the two stories collide. This is the convergence, two totally different designs that, according to this analysis, risk leading to one shared, dangerous outcome.
Let's put them right next to each other and see what's going on. Both platforms find a way to suppress our crucial interferon response. One uses a chemical cloak, the other uses biological hijacking. And both platforms end up pointing the immune system toward the exact same high-risk RLFRK sequence. One does it by presenting the whole protein, the other does it by zooming the attack right in on it. You see? It's two completely different roads, but this analysis suggests they both lead to the same destination, a potential autoimmune attack on the very systems that protect us from viruses and keep our nervous system intact.
Okay, let's pull all of this together and look at the big picture takeaways from this comparative analysis.
It really boils down to three key points. First, interferon is the primary target. Both platforms used really sophisticated methods, one chemical, one biological, to shut down our body's most important antiviral alarm system. Second, RLFRK is the common weapon. Both designs, in their own way, end up directing our immune system to attack this specific peptide, which creates a huge risk of autoimmunity due to molecular mimicry. And third, the potential result of all this is a real double whammy, a crisis on two fronts, with the possible collapse of our antiviral defenses and damage to our central nervous system at the same time.
So in the end, this whole analysis leaves us with a really big question to think about. When two completely different, smart engineering philosophies risk ending up at the exact same harmful destination, what does that tell us about the unintended consequences of trying to rationally engineer and manipulate something as complex as the human immune response?